Welcome to the Mucosal Immunology podcast, brought to you by the Society for Mucosal Immunology and Mucosal Immunology Journal. Each episode will feature in-depth interviews, discussions on the latest scientific research, and real-world applications that are transforming healthcare today. Available on all major podcast platforms.
Now for our mucosal research roundup, back again is Catherine Knoop, here to share the latest at Mucosal Immunology. Hi, Catherine.
Hi, Marian. The first issue of the year is available online, and I wanted to highlight a few of the great articles we have. Let's start with the literature reviews. First up is a reappraisal of IL-9 in inflammation and cancer from Martine Schoes and colleagues at Ghent University in Belgium.
Oh, I don't often think about cancer, but when I do, I don't relate it to IL-9 of all cytokines. What's the scoop?
This review may be for you then. There is mounting evidence IL-9 can promote tumorigenesis and even suppress anti-tumor responses.
That makes sense, right? Since IL-9 is traditionally an allergic type cytokine?
Yeah, the authors also discuss IL-9 in the context of asthma and allergic airway inflammation. This review ties these topics together by focusing on the sources, functions, and targets of IL-9.
You know, the review How the Early Life Microbiome Shapes Immune Programming in Childhood Asthma and Allergies really caught my attention.
Mine too. So this is a review from Stuart Turvey and colleagues at the University of British Columbia in Vancouver, Canada. This review highlights how the microbiota develops in early life and how dysbiosis can be linked to allergic diseases.
Do they connect this to microbial metabolites?
You know it. They discuss short-chain fatty acids and a few other metabolites that are important for the development of tolerance. Shifting over to our primary manuscripts, I really liked pro-inflammatory NK-like T cells are expanded in the blood and inflamed intestine in Crohn's disease. The team used mass cytometry or CyTOF to characterize T cells in the blood and intestines of patients with Crohn's disease. They identified Treg subsets in the intestine that can actually make interferon gamma when we're stimulated.
Oh, so these Tregs may be promoting the disease?
Yeah, it's still unclear if these are causing the disease or a consequence of the intestinal inflammation, but this is a fantastic dive into human T cell subsets during Crohn's disease. Finally, work from Ann Mueller and colleagues show IgA facilitates the persistence of mucosal pathogen Helicobacter pylori.
Uh-oh, what? The persistence? That doesn't sound too good.
Exactly. So strikingly, the authors found IgA deficient mice controlled H. pylori infections better than wild-type mice.
But doesn't H. pylori induces IgA responses?
Yeah, it does, but the authors hypothesize that IgA coding may actually prevent complement-mediated killing of H. pylori.
Of course, we can't forget our classic clearance pathways. Thanks, Catherine.
You can read all this and more at mucosalimmunology.org, home of the Mucosal Immunology Journal, the SMI-sponsored journal dedicated to all things mucosal. You can find reviews, primary research, and research collections from our entire archive, all available open access. Are you ready to submit your research to Mucosal Immunology? Your format-free initial submission will be given a first decision by an editor in less than 1 week. If you have a shorter story to tell, try our brief reports with up to 4 figures and a combined results and discussion section. Submit today on editorialmanager.com/MUCIMM.
Welcome back, Marianne. Ready to dive into another mystery?
Oh, I'm always ready, Ashu. But first, do you remember last week's puzzle? I know it had everyone thinking hard.
Oh, you know I have a goldfish brain. But I hope you remember, Marianne. We had tons of responses. And you listeners sure love a good brain teaser.
You bet we do. This one was a real challenge, but hey, it's time to announce our lucky winner. As usual, Catherine Knoop joins us from the Mayo Clinic, Rochester. Drum roll, please.
Congratulations to SMI trainee member Josie Muskie from Mayo Clinic in Minnesota. Thanks for listening.
Awesome. Big congrats to our lucky winner. You cracked the case. It is always exciting to see how many of you are getting involved in these historical puzzles. So the answer for last week's puzzle was the stranger. While the others were weakened by the disease or conventional approaches, the stranger's unique knowledge and isolation protect him from further contagion and give him the best chance of survival.
Yeah, I didn't want to isolate during COVID, but it may have saved my life, right? Okay, let's move on to today's mystery. This one is from the 16th century, and it is a nasty one. We are talking about the Great Pox, a disease sweeping across Europe.
Oh, the Great Pox sound ominous, right? The symptoms are terrifying. Fever, painful sores, and in the worst cases, disfigurement and death. The doctors are totally baffled. Some even thought it was a curse from the heavens.
Oh, yikes. But one physician, let's call him Doctor Who, is starting to notice a pattern. He sees that the disease seems to be spreading particularly among soldiers, especially those returning from war.
Exactly. And here is an interesting clue for our listener. In fact, the name of the disease might ring a bell. If you ever read poetry, as Doctor Who was also a poet, and named the disease after a boy who got punished by the god of sun. Pretty intense,
Ooh, I like that little clue, Ashu. So Doctor Who is onto something, but can you, dear listener, put on your 16th century medical thinking caps and figure out exactly what is going on here?
Listeners, can you figure out the disease? And who is this mysterious Doctor Who? There is a big clue in that famous poem.
Time to dig deep, detectives. The first person to send in the right answer gets a shout out in the next episode. We can't wait to see who cracks this one.
All right, Fox, get those thinking caps on. Let's see who can solve the Great Fox mystery.
Grab your lab coat or your driving wheel or a cup of coffee. I am Marion Brunk, and with me is Agnes Garcia, your host on the Mucosal Immunology podcast.
Today we have Dr. Cecilia Johansson. She's a professor in Mucosal Immunology in the section of respiratory infections at the National Heart and Lung Institute at Imperial College London. Hi, Cecilia. Thanks for being here with us. How are you doing today?
Hi, thank you for having me. I'm great.
Well, I think we will first start with asking you a little bit about your research career and your career path. So you are not originally from London, right?
No. I am Swedish, and I did all my training in Sweden in different places. And then I moved to the States and did a postdoc at the NIH in Bethesda, and then I moved to London and Imperial College.
Great, Cecilia. So you and your group recently published a paper on prior infection, bacterial infection, regulating or incrementing the severity of RSV infections. That was published in August last year at MI. This was a very interesting paper to read. It really emphasizes the double-edged system that is the immune system, right? It helps, but it also could be a little bit detrimental to the patient. Can you tell us a little bit more about this paper?
Yeah, so we are, the lab in general is interested in what drives severity after respiratory viral infections. And we know it can be a lot of different things, but it was very clear, especially during the COVID pandemic, that some people are completely asymptomatic and some get a really severe disease. And sometimes there are no obvious underlying situations that would explain this. So in the lab in general, we're very interested in finding out more, especially what the immune system does and how the mucosa can be potentially primed in different ways and react to the virus in different ways, good or bad. So what we had observed is if there's a neutrophil signature in the nasal mucosa, then that leads to more severe disease. And we found that both in a human challenge study using volunteers that got RSV and also in murine models of RSV. So for this particular paper, we then took that forward to start to kind of mechanistically look at what can drive more severe disease. And we use different PRR agonists and bacterial infection to see how that would change the immune responses in the lung mucosa before and then during RSV infection.
Yeah, that was really cool. So about your neutrophil findings, it was really interesting to me that depleting neutrophils did not help with getting rid of the infection, right? The viral load was not decreased. Was decreased with this. Is that correct? So I would have thought that neutrophils, unlike monocytes in your paper, were contributing to getting rid of the virus, but that was not the case in your model. Is that correct?
That is correct, and it is quite surprising. But if you do look through the literature, it's actually not clear what neutrophils do during viral infection. They are very important for bacterial and fungus infection. Neutropenic patients have a much higher susceptibility to bacterial or fungus infection, but not so much to virus infection. And we've done previous studies where we have focused only on RSV and depleted neutrophils and have not seen really any effect on viral load on disease severity. However, you always have to keep in mind that if you deplete a cell type, you take away both good and potentially bad aspects of functions of that cell, and timing is also extremely important. So I think there is more to do there, but we were surprised that removing the neutrophils during our kind of pre-exposure model, where we attract a lot of neutrophils, that didn't change the severity of the RSV infection, because we have a previous study where if you only have neutrophils there, it does change the severity of infection, but slightly later during the infection. So there is clearly some cellular crosstalk, and the exact combination of mechanisms that are running at the same time that will determine how the subsequent viral infection is received and controlled.
Mm-hmm.
That's very interesting. And I also have a question on that, because I was also surprised with the viral load. And then you show when you deplete inflammatory monocytes, right? New coming monocytes, then the viral load increases significantly. So that monocytes, as you mentioned in the paper, partially help on controlling the viral load, but not really a big change, right? So what would be the mechanism of controlling that viral load, and how important would that be to actually have a less severe immune response?
So the viral load is an interesting aspect and obviously important to control. So we have, not so much in this paper that we are currently discussing, but in a lot of our previous work, we have focused on type 1 interferons. And type 1 interferons, as all interferons, are extremely important. So if you remove the whole interferon response, you're going to have a much bigger influence on viral load. But then we have also shown that monocytes have an antiviral effect, which has been quite unappreciated, I think, in the field. Come back now during COVID a bit, but they are also important effector cells. And then NK cells obviously are also really important. And then you have the T cells. And I think if you remove any of these, you change the viral load a bit. But what it shows is that you need several mechanisms that happens at slightly different times that keep the viral load down. So if you remove one of them, you change the viral load, but at least in our models, not extremely dramatically. It's still controlled, but you see a shift. More viral load gives you more immune responses, usually, by default, and then the whole thing is rolling and gets worse in both the virus side and the immune pathology side.
I also read a bit on the papers with the Taiwan interference, and this actually drives me a little bit into what about dendritic cells? Have you looked at them too? Because you see how the different populations produce the main cytokines that you have seen that they are involved in that severity, right? And it's neutrophils play an important role in inflammatory monocytes, but you also show that there's the alveolar macrophages, right? Because there's a resident cell that first has this response, so then attracts those cells. And then I wonder, with being RSV, of course, interferon response because of a viral infection, what about dendritic cells?
Yeah, so the Taiwan interferons we have shown is mostly produced by alveolar macrophages. Dendritic cells are obviously very important. Probably not as an effector cell in the early antiviral response because they are so few and they leave, right? But really important in driving the T cell response, which obviously it's important to clear the infection in the end, but also to drive a bit of the disease, because actually the disease that we notice, both in mice and humans, is timed with the T cell response. So it's the kind of delicate balance of your magnitude of T cell responses and the disease severity. So the DCs will be very important, and I think they will be part in regulating the magnitude of the T cell response. And then not only that, because they will also regulate what kind of memory we have, and there is more and more focus now on looking at tissue resident memory cells with all infections we had and vaccines that are given intramuscularly. And we know that the early events that gives the signals to the DCs and the timing of those signals are really important in also determining memory T cell responses. So for example, if you have less interferons, we have shown in our system that you have a good T cell response, but you don't have a good tissue resident memory cell response, suggesting that you have a whole set of signals that needs to be completely coordinated and can influence different aspects of even a T cell, right? So it's really important to think about how these cells interact and when they interact. So yes, DCs are very important, but maybe not in these early events in terms of direct antiviral mechanisms, more like messengers and initiation of the T cells, which will be slightly later.
So you're mentioning the timeline quite a bit, Cecilia, and this is really important and really difficult to wrap our head around, in this complex context of bacteria and virus and the host and everything that happens. In your paper, you prime, let's say, your model with LPS or bacteria 12 hours prior to your challenge with the virus. Those 12 hours, how do you think this could translate to humans? Do we need to have this priming short before a possible infection? How does this work? Are we primed all the time?
Yeah, so I don't think so, but it was obviously the model that we went for because we wanted something that was highly controlled, and we also wanted to pre-expose, to kind of have a good response to the pre-exposure at the time of infection to really kind of answer our initial question. With humans, it's going to be very, very different scenarios. You could have a low exposure chronically, or you could have something that is more like a co-infection, which could then be shortly before. Or so I think it's the status of, in this case, the lung mucosal lining that could potentially be primed very differently depending on what situation you are, that then receives the virus in a different way. So there are some studies, not from my lab, but some studies that have suggested that children that grow up in a, or are in a very high LPS environment, they have a higher risk to develop more severe RSV infection. So that would be, you know, a more of a chronic exposure, and you know what goes on in their mucosa. It's hard to know at this stage, but we can do much more models in animals to figure out timings of events and what type of challenges, you know, if you have allergens or environmental LPS or microbiota, or, you know, there are so many things that could be altered to then change how the virus is received. So yes, this is one way of looking at it, but I think in the human scenario, we could easily add 20 new scenarios that would be worth modeling. So yeah.
Surely, yes. Thank you.
I think the paper shows beautifully, as Marion said, that the timeline really. I'm focusing on that, how the different cells, right, that crosstalk, and so how complex it is really. We cannot base it on one cell. And then talking about the model, I think it's a beautiful model you show with different PRR agonists with the bacteria and so on, and how you see the similar response. And then thinking about how difficult it is to really model the human setup in animal models. One thing that maybe can be a little more controlled, I don't even know if it can be, but it's at least the age of the immune system, maybe one could say. And I wonder, because that you did that in adult mice, right? And then we see, correct me if I'm wrong, that this severity is happens in young children and also in the elderly. And we know that the immune system is quite different. Of course, within different, it's like personalized, but these age groups, right? So have you tried to do the same very well characterized model? And maybe it's a question that you are now ongoing. But have you tried to do that in mouse pups or in aged mice and see? I don't know if the pups and the aged mice can really mimic better the immune system of those human patients. Have you tried that? Is that in the plan? Is there any difference we can already know?
Yeah, so we haven't tried this kind of pre-exposure model in the different age groups, but we know that elderly mice get more severe RSV infection like humans. So obviously there is that's something we really want to try, is what happens if you then expose them to different things. How much sicker would they get? And then neonatal mice are also really interesting. We haven't tried them. Overall, they don't get super sick with RSV, so it's slightly more difficult to model this. However, I should say that we haven't tried the pre-exposure model where things might be different. But neonatal mice is not like human babies, and that they get much sicker than adult mice. They do gain weight and all of that. But it's an interesting and very relevant question, what age does to respiratory infections, but also to this, how different co-infections or different tone of the mucosa can change. So very important.
Okay, so cytokines blockage had no impact on viral load. Is this something that is surprising? And in seeing this, how can we translate to potential treatments if cytokine blockage, and you have to block both at once, right? To ameliorate this weight loss that you see in the mice. So where do we go from knowing this to translation in terms of cytokines, if there is anything?
So it's not cytokine blockage, because if you block interferons, you have a huge impact on your viral load. So it's blockage of the IL-1 and TNF in this case. And I'm not so surprised that that doesn't influence viral load, because you have so many, your interferons are intact, and we look very early during infection. So the effect that we kind of expected would be on the immune response. It's interesting to then think, okay, that you're never going to go and necessarily block all type of cytokines in a human, but there is, for example, a lot of IL-1 receptor agonists that are already tested in autoimmune disease. Not saying that that will be to every patient, but maybe if someone that is more susceptible to repeated infections, or have had severe infections, or have had underlying conditions, maybe there would be room for that. I think at this stage it's so early, and our data, it's more suggesting that these cytokines that are from our bacterial stimulation or pre-exposure, and then together with the viral infection, is probably reaching a threshold that then gives more symptoms. So we need to understand much more what that threshold is and how that is regulated, and potentially find other treatment avenues than a cytokine, but in that kind of pathway. So I would say it opens a door more than define something very specific.
Sure, that makes sense.
So there's a difference. You clearly see a difference between IL-1 alpha and IL-1 beta. Why is that?
Yeah, that is something that has puzzled us, and it's still puzzling us. Two cytokines that work on the same receptor. So we were quite surprised. We don't have a definite answer, but the way we have thought about it is maybe the cellular source. IL-1 alpha is regulated different than IL-1 beta, and in terms of inflammasome activation and all of that, I think IL-1 alpha is also more pre-stored and ready to go. So it might be a timing event. Again, I'm just speculating because we haven't confirmed any of this, but it could be that IL-1 alpha gets released much quicker and have a faster effect, while IL beta is then coming later on. And what we have noticed in this model is that everything is happening really fast. So maybe again we come back to timing, that it is a timing difference. But it could be many other things. The cells that produce it, but we were surprised.
I was wondering if you further phenotyped those neutrophils. There is a recent paper that came out showing that CCR3 expression in influenza infections was related to severity, recruitment of more neutrophils, more activation of those cells, etc. Is there anything that you've looked at in addition to the expression of those IL by neutrophils?
We haven't really, no. So the answer is no. We haven't looked at those receptors. It will be really interesting to see if it's different activation status. We see that the the neutrophils get activated, but we have more kind of focused on the functional readouts. So it will be really interesting to kind of look into more of the specifics of the neutrophils and how potentially their lifespan increases, and those aspects that we haven't really done that yet, but that's something we want to do.
Just a quick question with not knowing at all, but the RSV is known that when it can cause a severe phenotype, at least in early life, then it these kids are more prone to develop asthma. And I wonder if the neutrophil response can actually, and the severity of the response led by the neutrophils can actually is related to their function. Is it that, asthma
You
related
mean
to neutrophil?
the link to asthma?
Yes.
Hard to say. Children with RSV bronchiolitis have a lot of neutrophils in their airways, lots and lots. That could be due to many things, and they might be there because of the infection, but actually not doing much to the infection, because this is also usually much later on when the children are actually presented in the clinic and not these early stages. They can cause a lot of damage, and we know that these children's lungs are quite damaged. So can other cells, so can the virus. And all of that combined probably leads to healing process and then subsequent issues. But there seem to be so many different aspects that then leads to these children and their kind of lung health later on, that is harder to say what it is, and maybe it's even different for different children. And we need much more research in that to see the long-term consequences. We're probably going to learn quite a lot from the severity in the COVID patients and how they heal the lung and the sheer number of people that had really severe infections. And we see more and more studies coming out on that, describing different factors leading to subsequent lung health. So yeah, part of the puzzle, but it's hard to say what the whole picture is.
Thank you, Cecilia. And finally, I think you're also working on how viral infections also influence lung cancer. So talking to that other aspect as well, is there anything that you can comment regarding RSV and the severity of its infection on lung cancer? Is that have you looked at that?
Yeah, so we have for quite a while been interested in how the two diseases, so viral lung disease and lung cancer, influence each other. So both how the lung cancer would influence the course of the infection, or also how lung infection influence cancer growth. So we have several models, both looking at metastatic lung cancer and primary lung cancer. And we've been mostly focusing so far on metastatic cancer after lung infection, because we know that the lung infection is changing the lungs quite dramatically. And what then happens to the lung being a new home for metastatic cancer. And there is a lot of crosstalk between these two diseases, and it's really, really interesting. And again, comes back to a lot of timing, that depending when things happen and what kind of immune components that are active, it also change both metastatic spread and cancer growth in the lungs. And we use these models to try to learn more about which mechanisms are good or bad for the host in order to combat cancer. So that's a slightly different angle in the lab, but really, really interesting. And obviously the lung, as all of our mucosal sites, are often, battling several things at the same time. So yeah, a different question, but very interesting.
Yeah, just to make a comment on that, I think it's beautiful to do that work and also expand, because at the end, I think we realize that immunology is very much in all diseases, right? And also within the mucosal sites, that response that happens there, whether infections or cancer, right? That local site is quite important, and we see that actually cancer, it's normally like this is a low-grade inflammation, right? Or inflammation that hasn't developed, and then there's this sort of a tolerance or so. So definitely a lot of things in common. And I think it's super interesting and actually a lot in common, I would
Yes.
say. I worked in cancer too, and I think there's so much so much in common.
Yes, and we talked a lot about timing, and unfortunately, I think our time is coming to an end. It was so lovely talking to you, Cecilia. Thank you so much for your time and your interest in coming on to the Mucosal Immunology podcast.
Oh, thank you very much. Thank you for having me. It was a pleasure.
Thanks for tuning in to the Mucosal Immunology Podcast. The Mucosal Immunology Podcast is produced and brought to you by the Mucosal Immunology Journal and the Society for Mucosal Immunology. Thank you to our content contributors, Drs. Marian Brunk, Agnes Garcias-Lopez, and Ashu Mangalam. This podcast is produced by Matt Close and Catherine Canoop. We hope you enjoyed this episode. Remember, the journey of discovery in mucosal immunology doesn't end here. Stay connected with us for more in-depth discussions, expert interviews, and the latest research breakthrough. If you found this episode valuable, please subscribe, rate, and leave us a review on your favorite podcast platform. Your feedback helps us reach more researchers passionate about mucosal immunology. If there are questions or topics you'd like us to cover in future episodes, we'd love to hear from you. Connect with us on Blue Sky, LinkedIn, Facebook, or Twitter/X. Thank you for listening.